So good morning. So I'll speak about some of our recent work in evolutionary biology. And this work is done with Archana Devi, who's currently a postdoc at Arizona State. Okay, so we are thinking about a population which is currently well adapted to an environment. So here, well adapted simply means that it has a high fitness. So now, suppose environment changes so that the population is no longer adapted, then it has to evolve and then get adapted to this new environment. And the general question is, what are the dynamics of this adaptation process? So dynamics, these adaptation occurs at both phenotypic and genotypic level. So genotype, I think all of you have heard this word. So it just simply means at the genetic level. But phenotype is simply some visual characteristics. For example, the coat color of the mice or the skin color, hair, and so on. Okay. So in general, adaptation occurs at both what you can see as well underlying genetics and underlying DNA level. However, the current descriptions in evolution, they operate only at one or the other level. So there's one standard description which goes by the name quantitative genetics, in which one imagines that a phenotypic trait is controlled by a very large number of genetic loci, genetic sites. And when environment changes, then the frequencies change by only tiny amount. So there's a tiny amount of change at each site, but at a large number of sites, so that there is a finite change, however, in the phenotype. Okay. So in this picture of quantitative genetics, because there's too many loci involved, too many sites involved, one simply ignores the genetic details and works only at the phenotypic level. Other description is called the population genetics level, in which one works only at the genotypic level, and one assumes that when the environment has changed, then mutants will arise continually. So one mutant will arise, and if it escapes stochastic laws, it might spread through the environment. That means the frequency can change by a large amount. It can even become 1. So in contrast to quantitative genetics, where there were tiny changes, in population genetics picture, adaptation is through large changes in frequency. However, typically in population genetics, you don't connect things with phenotype. So clearly, for a complete description, for a complete theory, one should integrate both phenotype and genotype into a single framework. So ideally, one should start with phenotype, and for example, one can specify fitness because the reasoning being the selection acts on phenotype. So for example, predator, it will see the prey, it will see its color, wing color, and so on. It will not see its DNA, right? So therefore, one should actually start at the phenotypic level, use the phenotype-genotype map to go to the genetics, work out all the evolutionary dynamics at genetic level, use this map and go back and complete the circle to make predictions about phenotype. So this is clearly a challenging problem, and people have thought about it for some time, but there has been the progress only in recent times, because now we know a lot more about genetics due to genetic sequencing, and we know a little bit more about this map. So I'll tell you about our effort in this direction. So our model is for a phenotypic trait for which there's an optimum. So for example, you can think of a height. There's the optimal height. So we're assuming, so very tall or very short individuals have a low fitness, and there's some intermediate height for which there is a moderate fitness. And one can model this by, for example, a Gaussian. Then each individual is uniquely specified by its genotype. So I'll give you a one-to-one map, which is a genotype to phenotype map, for which we can make some reasonable assumption, simple being that it's an additive map. Let me explain this point. So suppose I have an individual whose genotype has on the first site, there's a tall gene which is on. At the second site, short gene is on, and so on. Tall gene is on means it increases the height by a certain amount. Short gene means it decreases the height by a certain amount. So we just add up all these with the correct signs, and there is some effect of the tall gene. It increases height by a certain amount, and short one decreases by a certain amount, and so on. And these effects at different sites can in general be different. So now we know how to relate genotype to the phenotype. I know the phenotypic fitness, and then I know something about the basic evolutionary forces and can run some stochastic process in some standard stochastic process in population genetics. So I'll give you a flavor of that, a standard process which is known as Wright-Fisher process. So it deals with n individuals, and there's a little cartoon of that. Suppose I have 4 individuals, each of them has a genome of length 4. So suppose this individual has a genome like TTT. It has a low fitness. As I said, we are thinking not of extremes. It has a low fitness where others are somewhat higher fitness. So it's unlikely that this will have any offspring, or it will have fewer offspring compared to the other ones. So that's the selection. Then, because each system has a finite carrying capacity, there's a random sampling to reduce the population size to the previous one, and there can be mutations between S and T and T and S, so that finally you wind up with this offspring generation. So there are several steps involved in going from one generation to another. You can think about it later, and you can put this program on the computer and run it. And the question that one can ask is that at site I, what's the probability? The frequency is xi, and more generally, you can ask for the joint distribution of these frequencies at time t. So you can run this program, but what we did was to look at a Fokker-Planck equation for this joint distribution. So one can write down this probability, and this is the standard one in which you have this drift term, and this is the diffusion term, but I'm thinking of a genome of L sites. So therefore, I also have this sum over for these operators. So we need not worry about these details here, what these M's and V's are. Only thing I'll point out to you is that the diffusion term at the IS site depends only on its own frequency, but the deterministic drift term is such that it depends on all the other frequencies. In other words, this is not an independent site problem. Okay, and the stationary state of this model is known for quite some time, and because of these interactions, the distribution is not a product measure. There is a multiplicative part, but there is also some cross terms in xi and xj. As I said already, this is not an independent site problem. Okay, so this is a pretty complicated thing. At least I can understand what it means. So what we did was to integrate over all but one frequencies to understand the marginal. When we look at the marginal distribution, we find that in certain parameter regimes, it can change shape in the following manner. There's a critical effect size below which the distribution is unimodal, and above it it's bimodal. And one can work out more details of this. But what we are interested in is the adaptation process. So let's come back to physics. So what we are thinking about is that I know the distribution, the stationary distribution. I prepare the state in the stationary distribution, and let's say the optimal height was some h naught. Now we change the environment, so the h naught has changed, and now I have a different system. Population is no longer in equilibrium, and the question is, what are the dynamics of these allele frequencies over time? Specifically, we're asking that at the site at which there's a bimodal distribution, does the change in these frequencies occur by a tiny amount? So remember, this is a bimodal distribution. So you can start at one mode and do teeny changes there, and therefore adapt and achieve the new equilibrium. So does it happen that way, which is the quantitative genetics picture in which changes happen by tiny amount? Or more like population genetics picture, in which you start at one mode, look at the blue one mode, but go to the other mode of the bimodal distribution, which is the population genetics picture. So is it this one or that one, or maybe a combination of them? So this is one very important question in the subject. Okay, so we can, to quantify this, we can write down this probability of making this large change. These are called sweeps. So that means we're starting at the higher mode site, and what's the chance, given that we started at the low mode, the population goes to the higher mode. Lower mode site are, one of them I know because I know the stationary distribution, I know this marginal, right? But to find this, I need to know the dynamics. We are dealing with a Fokker-Planck equation with a large number of sites, and they're interacting sites. So of course, we cannot solve that exactly, okay? However, if I'm thinking of large populations, which I am doing today, if stochasticity is small, then one expects intuitively that the dynamical distribution would be a Gaussian centered about the deterministic value, deterministic answer, which we had worked out a while ago. What I want to emphasize is that this Gaussian distribution is not an assumption. It's something you can show systematically from what is called Fan-Campus system size expansion. So for our purpose, that was sufficient. So we know this marginal, we know the Gaussian, we can put things together and work out. And what we find is, these are the results from our simulations versus our expressions. Okay, so this result for biologists is sort of
2
interesting.
more minutes.
Yeah, thanks. It's sort of interesting because previous work has shown that if the model is deterministic, that is population size infinite, or it's very small, then these large changes don't happen. Sweeps have low probability or negligible probability of happening. Our result shows that in the moderately large population in this parameter region, there's a higher chance. And it is useful in the subject to use these large changes to find out where and how much selection occurs in the genome. But to make a real connection to biology, much more is needed, which we at least have not done. In any case, I hope I illustrated to you a simple an example where we can use ideas from StatMech and theory of stochastic processes to learn something about of other in other problems. I'll stop with that. Thank you.